Hello, everyone. So yes, I'll be talking about circular Mars. I'm here mostly as the scientific director of the Melissa Foundation, not as not representing my work. Part of my work is part of the Melissa project. I will explain what it is, but Circular Mars is really about the Melissa project and how this can help us in exploring the red planet. But before going there, we'll start a little bit closer with a view from our own planet, and that's from the satellite that was placed at the Lagrangian point 1, and it has filmed the Earth for many years, and it has provided these images for over a decade, and sadly was disconnected on this October by the Trump administration. But my point is here to show you a living biosphere with a lot of ecosystems, a lot of closed geochemical cycles, everything at equilibrium with, despite that, dynamic processes, these ecosystems interacting, and this is what is providing us our environment. Something that's really important is that these cycles that provides us our environment are based on very long durations, very large buffers. When we go to space, when we go to Mars, we don't have that. I think all the presentations today have shown that. We'll go to some sort of desert where we don't have all the resources we have here, and we have to deal with very close spaces. So that means at the same time being far away from most of humanity, living in very close spaces, very nearby our other crewmates. But while we do that, we'll have to remember what we need per day, and that's just what we need to survive. We need oxygen, water, and food. You know that. We produce CO2, water, and dry matter. And all of that needs to be dealt with and provided. And so this is what we have life support for, and the main functions are the atmosphere management. And so I'm just not talking about recycling here, I'm mostly talking about providing these atmosphere composition we need, and removing particles and microorganisms that are not wanted. Also filtering volatile organic gas and really managing the atmosphere. Same for water. We'll need to provide enough water. We'll need to deal with wastewater. Food production, food provision. Same, we need to have enough food, and we need then to deal with wastes. And once we have done all that, we need to do that at temperature and pressure that are sustainable for human life. So that's the main functions of a life support system. And we can achieve all of that with different means. We can launch everything at once, we can resupply, we can recycle, and we can use Institut resources. Or, of course, we can also use a combination of the four. If we make a really quick computation, using only launching or resupplying for a 1,000-day mission on Mars for people, we'll compute what we need for just surviving. So metabolic consumables, that's water, oxygen, food. So it's about 5 kilograms per day per person. So you've already done the computation in your head. It's 20 tons. And if we add the hygiene water, we have 80 tons. And then if we add all of that, it's 100 tons. We've talked earlier today about curiosity and perseverance. That was 1 ton that we landed on the red planet. And you may remember the landing of Curiosity. It was, pretty impressive. It was hard, but we managed. Now imagine doing that 100 times. not really convenient, and this is why we need to recycle resources and really move from the approach we've used for decades on the International Space Station, which is launching and then using and then well, going to waste. We need really to use closed loops. And that's what the Melissa project is about. It is inspired from a natural ecosystem. So really the concept of a lake ecosystem where you have the degraders, you have the consumers, and you have the producers. And these ecosystem is compartmentalized into different boxes, and each of them have a specific function. And so we talk about an artificial ecosystem. And in the case of Melissa, so Melissa is the acronym for Microecological Life Support System Alternative. We have 3 microbial compartments, which are the degraders. So the first one will liquefy the wastes. It's based on anaerobic digestion. The second one is based on microbial electrolysis cell, and it's for the elimination of organic carbon. And then the third one is nitrification. So in this last stage here, we the aim is really to transform nitrogen into a form that's usable by plants and microalgae. And the plants and microalgae, that's our producer compartment. So it's a double compartment. We have here spirulina and here different types of plants. And they produce, with photosynthesis, food, water, oxygen, using the CO2 that's exhaled by the crew. And the crew in this ecosystem is our consumer. So that's the Melissa concept. You may have noticed, you may have noticed here that we have different organisms. We have bacteria, and then here we have microalgae, well, cyanobacteria, and we have plants. And then, of course, we have the animals here. All of that makes some difficulties because we need to first work at different scales. We need to look really at what's happening at the metabolic level, going to the microbiological level then, and then the process level, all the limiting factors we have, all the kinetics, and then the system level to be able to assemble and integrate everything. All of these systems, all of these compartments, each have their own time constant. Microbial organisms have really fast time constant. They will develop really fast. So we have a growth cycle that's rapid. Plants, it will depend on the species. If you are dealing with lettuce, you have a growth cycle of about 3 weeks in control environment. If you go for potatoes, it's more about 3 months. Humans, if you look at how, if you look at their breathing, so the gas loop, that's a really short time constant, because if you don't provide enough oxygen in a short time, you'll have your crew dying. The water loop, so the liquid loop, that's a little longer time constant. You can stay a little longer without water, and then food is longer. So you have, just for you humans, you already have 3 different time constants to deal with, depending on which loop, like liquid, solid, or gas, you're dealing with. And then you add all of the organisms. So it's really about integrating compartments with, which are moving at different frequencies. So we have an engineering approach. We develop knowledge models, predictive mechanistic models that are validated with experimental work. And once this is done, we can, in parallel, develop technologies based on these experiments, which have been used to validate our models. And we also develop predictive control, and we integrate. We integrate the compartments physically. I'll detail that later, and we integrate them also from a control level and with models, to end up at our systems and operations level. And so for that, we have first, we have to develop fundamental and applied research in different laboratories, and then validate, demonstrate technologies and processes in relevant environment, mostly microgravity, and also in operational environment in our pilot plant, in our ground demonstrator. The Melissa project, really briefly, is a project that's been around for 35 years. It has 16 partners. ESA is one of the partners, and so the European Space Agency deals with the management of the project. And so I was saying we have 16 partners. We are really gathered all over Europe. University Clermont Auvergne, so my university is one of the partners, a historic partner. We've been working for over 30 years on that, mostly on the modeling and simulation part. The project, or the program, I should say, because it's bigger than a project, is organized in 5 big phases. We have the ground research and development, which is happening in our labs. So that's what I was saying, really to have fundamental and applied research to characterize each of the processes that are used. We then have the flight demonstration and ground integration. So number 2, number 3. Terrestrial applications. I'll have a word on that at the end of this presentation, really briefly, but we'll mostly talk about that in the roundtable afterwards. And we have the education and communication part, and that's the part that is managed by the Melissa Foundation, and we have a PhD and postdoc program. Today, I'm going to present an example of one of our flight demonstration and an example of our ground demonstrator. So for the flight demonstration, we have flown twice a small photobioreactor to the International Space Station in 2018, 2019. That was in batch mode, and then in last year, 2024, 2025, in continuous mode. Really small photobioreactor, 50 milliliters with spirulina. And some of the results we have, so yeah, that's the flight hardware open so that we can see all of the different components. And here are some of the results obtained. It's the cumulative pressure of oxygen within the photobioreactor. The goal of this experiment was to demonstrate that we can have an extended culture of the spirulina, that we can produce oxygen, that we are able to deal with the gas and liquid separation, which is not obvious in microgravity, and that we can also fit our model to the experimental data obtained, our predictive model. So here you have cumulative pressure in different batches. The ones that are with dashed line are the ground batches, and the ones with the dots are the ISS batches. And so if we take the example of this one, for example, we have we will look at how the cumulative pressure is predicted by our model. So that's our model is the really small. Oh, I see it really small, but because I'm far. But that's the dotted line here, and the experimental cumulative pressure is the really dark line. Our model was able to predict really well the cumulative pressure on the ISS produced by spirulina in this small photobioreactor. For the biomass production, same, we have the experimental data here, that's the dots, and the model is the dashed line. And here again, our model predicted quite well the evolution and the biomass production from this photobioreactor. I don't have the continuous mode results yet, because it was the experiment happened last year. They're still analyzing and processing the data and in the process of publishing the paper, so I don't have them to present, but you can be on the lookout if you're interested. Moving on to the Melissa pilot plant. So the Melissa pilot plant really is the ground demonstrator. It's located at the University Autónoma de Barcelona. We have 4 compartments that are in pilot size. We have the nitrifying compartment, the photobioreactor, the food or plant production module. It's not really a greenhouse. It's really a container with plants inside. And then we have our crew. Our crew at the moment is composed of five rats. The reason why we have rats at the moment is because we needed to have a scalable system, and to have the respiration of 1 person, that would represent 40 rats, more or less. So it was decided to start with 5 rats and be able to size the system for that type of crew. Keep in mind here that we are not talking about a system that's designed for space. It's not like miniaturized for space. It's a system that is designed to show the to characterize each of these compartments and to demonstrate the integration on the ground, to be able to really follow each of the processes and go and do these a stepwise integration. So that's why, I mean, you see, you have the people for scale here. We are talking about really, really big systems. This is not intended to be launched to space like this. And you've seen the small photobioreactor I just showed you earlier. So at the moment, we have 4 compartments that are integrated together on the gas and liquid loop. We have, so why gas and liquid loop? The integration is really done step by step. First, gas integration. So the different compartments are linked together on the gas loop. And the idea is really to manage the oxygen need by the crew and see if we can adjust these needs with the photobioreactor. That was really the first step. And then on the gas loop, the greenhouse was added. The photobioreactor enables to deal with the really fast demands of oxygen, because the spirulina can react very quickly. And so if there is a high demand, if, for example, if we don't have a rat crew but a human crew, if our humans are running, for example, if they are having their daily exercise, they will need more oxygen. And then a photobioreactor can deal with these very brief variations, while our plants here will deal with the long-term variations or needs of oxygen and the scrubbing of CO2. So at the moment, our crew here is breathing the oxygen produced by the plants in the photobioreactor. And they are providing these photobioreactor and these plants production module with carbon dioxide. At the same time, the nitrifying compartment is fed with urine. So first synthetic urine, and I think in a few months, it's either now or in a few months, they will start experimenting with human urine. And so human urine or synthetic urine is degraded here in the nitrifying compartment, and this is fed via the liquid loop to the photobioreactor and the plant production module. So that's the status of the ground demonstrator. Now, briefly on the terrestrial applications, I don't want to take too much time here. I really just want to tease you for what we're going to discuss afterwards, but then you have the images of the systems I might be talking about. Everything I've just presented really has applications for more sustainable technologies on Earth. And when we look at the Sustainable Development Goals of the United Nations, we can answer a few of them. On the grey water recycling, for example, we have a demonstrator that's been installed in the Concordia station, well, it's not a demonstrator. It's a working it's a working facility. So it's been installed here for about 20 years, and it's recycling the shower water. We have had a recycling system for the showers at the Roland Garros tennis tournament since 2020. It's recycling the shower water into the toilet flush, and so just briefly, it's just it has to be disinstalled each after each tournament, because in France we cannot have such recycling system in place for well, long term. It can be only temporary. Yellow water recycling. At the ISA headquarter building, we are collecting urine. So there are some toilets are equipped with direct urine collection. This urine is separated between water and all the urea and nitrogen and that we need, and it's converted into an NPK, so nitrogen, phosphor, magnesium fertilizer, orin, that's actually commercialized, and the water is reused in the toilet flushes. We also have another technology. So here it's based on a bioreactor. This technology here was developed at the University of Ghent, and it's using electrochemistry to also recycle urine, and by local recycling, so collecting really on site and then separating water from urea. Last application, we can collect CO2 from a cement plant, for example, and valorize it with biomass, so biomass production. So here we have this project called Cement Algue, which is basically, that's the biggest cemetery in France. Next to Lyon, CO2 is collected and used to grow microalgae. And then microalgae can be used for either food or fuel production. Well, I'll just go very quickly on the conclusion, but yeah, if we want to go to Mars, we'll need to regenerate our resources. We can only produce food with biology. So we'll have to look into bioregenerative life support systems. Melissa is an Earth-inspired ecosystem, but follows really an engineering approach. It is based on ground research and development to enable us to demonstrate our processes on the ground, but also in space. We've had a successful flight experiments with a photobioreactor, small, but it's working in batch and continuously. We have a dedicated ground demonstration facility in Barcelona. Integration is in progress for the liquid and gas loops, and numerous terrestrial applications for a sustainable future. I was asked very briefly to advertise this. If you're interested in space biology, sorry, it's in French, but if you're interested in space biology, you can scan this QR code or write to this address. This is a master's program opening in September 2026, Master M2 Biospace Expert. Yeah, and I'm just waiting for you. Thank you.
Just one point, and we are both lecturers in this
I was not a lecturer. Well, I'm not a lecturer. I reviewed some of the applications for some, or if I'm a lecturer, I'm not yet aware. But okay.
Okay, I've seen your name as a
thank you. Okay.
teaching. This,
so maybe I don't know.
Anyway, so yes, one question we ask, please.
One of the reasons why we don't see a lot of closed life support systems like this for future mass-spatial architectures is the risk of infections inside. That's one thing. Can you elaborate about the risk of introducing a fungi or something that is just crushing the system?
Yeah, that's a really good question actually, because recycling is increasing the risk, and most of the activities here the reason why everything is modeled, predicted, and investigated in detail and taking so much time is because we want to de-risk everything before flying in an operational system. Now, indeed, we could have a microorganism being introduced and crashing the system, but this is why we have redundancy in space systems. So the Melissa loop, and that's my personal opinion, is not intended to be flown as a standalone system, but as an integrated within a greater life support system with redundancy, physical, chemical redundancy for oxygen production, for water production. For food, it's not possible, so we'll have to have multiple different systems for growing that, but we can use different organisms. Here we are based on spirulina and plants, but we can imagine having mushrooms, having aquaponics as well. There are multiple projects on that. So it is possible to really deal with these, to deal with this risk with just having more redundancy and being prepared by really knowing exactly what can happen. I know we cannot know 100%, but yeah.
Last question.
Yeah, how fast can your bioreactor accelerate, for instance, the production of oxygen with spirulina? What's the time?
It's instantaneous.
For instance, to double the oxygen.
You
feed it more. You feed more spirulina, or
it's their activity. Of course, you have the limit of photosynthesis,
right?
but it's really you increase the light, you increase the oxygen production right away.
So it would be the light that you increase.
Yeah.
Okay.
I mean, if that's for like fast regimes. If you want to, if we talk about an intensified photobioreactor, you at some point you will have to you have a trade-off between light and volume, and so either you increase your volume, either you increase the light. That's for the sizing, but then for a rapid increase, you increase the light, you increase your oxygen production to your limit of your size. Yes.
So thank you very much. We will discuss this longer, I think,
Yes.
during the discussion panel in about half an hour.
Yes.
So last talk before the discussion panel, Lucie Thibault from University of Bremen. But she will come back. She will come back in a couple of minutes.